Well, hi, you guys. Thank you for joining today for our ICRE Expert Series talks. I am super excited to have you guys today because yesterday was World Wetlands Day. So today we're here to celebrate World Wetlands Day to bring awareness of their importance in wetlands. And for those that don't know, I don't know if Marjorie's going to be talking about this, but wetlands are a vital habitat for a lot of plants and animals, especially a lot of endangered species. And they also provide a lot of human services for us. And so that's why it's very important for us to be able to conserve the remaining wetlands that we do have. And for today, I have the honor of having Marjorie Mednikov. I met her as an undergrad at Cal State Long Beach, and I'm just so excited to be able to hear what she's been up to in her PhD program. So Marjorie is a second-year PhD student in the marine biology program at the University of New Hampshire. Her research focuses on evaluating restoration techniques that can enhance salt marsh resilience to sea level rise in New Hampshire and Maine. Marjorie received her MS degree in biology in 2019 from Cal State University of Long Beach and worked as a natural resource field technician and an environmental consultant before beginning her doctoral program. So with that, I would take Marjorie, you could go ahead and take the stand.
Awesome. Thank you so much, Anita. Good morning, everyone. Thank you again for joining in. As Anita mentioned, my name is Marjorie Mednikova, and I'm in the second year of my PhD program at the University of New Hampshire. And specifically, I'm in Dr. Greg Moore's lab. So today I'm going to be speaking about building resilience within salt marsh habitat, and specifically highlighting some opportunities for innovation and collaboration in marsh restoration efforts. So I want to start off by wishing everyone a slightly belated Happy World Wetlands Day. This slide has a collection of photos from several different types of wetlands, including cypress swamps, mangrove forests, salt marshes, and bogs. While each of these wetlands has a unique role and different defining characteristics, all wetlands are ecosystems that are defined by the influence of water on soil and vegetation, and they have a lot of overlap in the functions they perform, as well as the services they provide. And I'll go into a little bit more detail on these services because the 2024 theme for World Wetlands Day is wetlands and human well-being. So in the first part of my talk, I'm going to be addressing the importance of wetland habitat more broadly, sort of expanding on the introduction that Anita gave, in addition to some of the implications of wetland habitat loss and priorities for restoration efforts. I like to begin any talk about wetlands with a reminder of the many valuable services that they provide. Wetlands play key roles in biogeochemical cycles, as their microbial communities are important in nitrogen cycling, and their sediments store carbon, which is an increasingly essential service as global temperatures rise. Wetlands also provide structured habitat for fish, birds, and invertebrates. Many fish, including commercially important species, seek refuge from predators or spend their juvenile stages in the sheltered habitat provided by coastal wetlands, such as salt marshes or mangrove forests. Wetland habitats, especially in coastal areas, are valuable buffers between land and water, filtering runoff from the land and attenuating or reducing wave energy from the ocean as it meets the shore. Each of these ecosystem services has economic value. So a 2012 study by De Groot et al. Estimated wetland services to be valued at approximately $193,845 per hectare per year. And it's important to note that this was in 2007 dollars, so this valuation is a conservative estimate, especially considering the amount of inflation happening today. It's also important to note that while we can estimate the economic value of these services, the ecological roles of wetlands are inherently or intrinsically valuable in a way that goes beyond dollar estimates. Tying these services back to the World Wetlands Day theme, many are integral to human well-being. Wetlands have traditionally been and currently remain important sources of food for humans. Fish and shellfish are a major component of human diets and are important for the livelihood and subsistence of coastal communities. Wetland plants themselves are also an important human resource. For example, the top image on this slide depicts the harvest of wild rice by the Ojibwe people in the Great Lakes region. In addition to providing resources for direct human consumption, wetlands also improve water clarity and quality for human health by filtering pollutants and sediment from the water within them. Coastal wetlands are also important in protecting coastal communities from storm surges, as the roots and shoots from vegetation reduce wave energy and can also reduce flooding. This is an especially important role as increased storm intensity is an effect of climate change. And you can see in the bottom photo, which was recent flooding observed in coastal Maine, that this can really affect coastal communities. Finally, humans enjoy wetland habitat for its recreational and its aesthetic value. And wetland habitat provides ample opportunity for kayaking, bird watching, fishing, and enjoying trails within wetland preserves. The many benefits of wetlands, especially for humans, are important to keep in mind when considering the impacts that humans have on wetland systems. So despite what we now know about the value of wetlands, they were not considered to be useful ecosystems during the period in which the US went through substantial population growth and industrialization. Wetlands were extensively drained and converted for agriculture or for residential or industrial development. This resulted in an estimated loss of 53% of the total wetland habitat in the US between the 1780s and the 1980s, and this loss does continue today. Wetland loss has varied by state, as you can see from the map on the left. Over 90% of California's wetland habitat was lost during this time period, especially along its extensive coastline. And freshwater wetlands in the Midwest also saw substantial losses despite their inland locations, as you can see from the cluster of red states in the center there. While New England has seen a somewhat proportionally smaller loss in habitat, it still experienced a loss of an estimated 37% of salt marsh habitat since the 1700s. Despite this concerning loss of habitat, in recent decades, the US government has implemented protections for wetlands, including the Clean Water Act in 1970. Section 404 of this act regulates dredge and fill within wetlands with the overall goal of reducing and mitigating impacts to wetlands and waters of the US. In addition to regulatory protections afforded to wetlands, there are a wide range of habitat restoration efforts around the US, many of which might be well known by the people in this call. Wetland restoration techniques can include invasive species management, such as removal efforts for the yellow flag iris or the common reed, plantings of native species, including dune grasses, mangroves, and salt marsh grasses, shellfish restoration, including eastern oyster reef restoration in the Chesapeake Bay, as well as Olympia oyster restoration along the Pacific coast. Hydrological restoration, including removal of tidal restrictions or filling ditches within salt marshes. And living shorelines projects, which use nature-based infrastructure to enhance coastal resilience. And these are just a few examples of the many types of wetland restorations that take place both in the US and across the world. With this diverse range of wetland restoration efforts across the country in mind, asking the following question becomes especially important. What are the knowledge gaps and research priorities that should be addressed next in coastal wetland restoration? As anthropogenic activity and climate change continue to alter our wetlands, what should we focus on in preserving and restoring them? So this question formed the basis of an interview project that I conducted in collaboration with Orange County Coastkeeper and the Honda Marine Science Foundation in 2020. And Christine is actually a co-author on this paper, so she'll be very familiar with this work. Our results were published last year in the Bulletin of the Southern California Academy of Sciences, and that document is linked in this publication or in this presentation, as well as on my current lab's website. So a small plug to check that out if you're interested. As part of this project, I conducted a literature review in addition to 30-minute interviews with living shorelines restoration practitioners, primarily along the Pacific coast, but within other US regions as well. And the locations of the interviewees from this study are depicted in red on the map shown here. Interviewees included biologists, ecologists, oceanographers, engineers, policy specialists, and stakeholder engagement professionals, basically people who have extensive experience with community outreach. I asked them to identify some of the major challenges and barriers in coastal restoration, with a focus on living shorelines projects, but with the mindset that this could be expanded to other wetland restoration techniques as well. I also asked them to identify some opportunities for new projects and programs that could be particularly useful in this field. Interviewees in this study identified a lack of long-term monitoring as one of the primary limitations in living shorelines restoration. This includes a lack of information on long-term success rates and durability of restoration projects, as well as information on maintenance costs and timeline from implementation to monitoring. They also identified a need for innovative demonstration projects or pilot projects that test new restoration techniques among different communities and in varied coastal settings, so from open coast to more sheltered environments. Practitioners also highlighted a need for more effective communication and data sharing between different organizations and communities, as well as a need for standardized monitoring metrics to better compare restoration projects between regions. There's also overall a need for better understanding of permitting requirements, which sometimes can be very complicated and actual restoration tech practitioners are not fully familiar with them, and a better need for communication with members of regulatory agencies that will be instrumental in getting living shorelines projects off the ground. Finally, interviewees also emphasized the importance of engagement with members of the local community, from educational initiatives to workshops with local property owners, who are often a very, very important group in getting permission to do coastal restoration work. To address these challenges, interviewees proposed various projects and programs that could be implemented or better funded moving forward. First, they encouraged investing in innovative pilot projects with scalable restoration techniques, so those that could be applied in a wide variety of coastal settings and permitted on a larger scale as well. Secondly, they recommended standardizing both restoration and monitoring methods, as well as replicating projects with an experimental design in different coastal communities. Third, practitioners suggested direct comparisons of nature-based or green restoration techniques, such as living shorelines, with gray approaches, such as shoreline armoring with bulkheads or seawalls. And in doing so, they recommend providing empirical data on the ability of native species or habitats to effectively stabilize coastlines as well as hardened structures. Other suggestions included training programs for engineers interested in nature-based infrastructure, communications campaigns that allow local communities to visualize the goals of these projects, and continued stakeholder outreach and community engagement efforts, especially in underserved communities that could benefit greatly from both a deeper connection with coastal habitats and the services that these wetland systems can provide. So the responses to this interview project were extremely insightful and helpful in identifying goals for future restoration efforts. So after gathering these responses, I was left with the following question: how can I integrate these take-home messages into my future restoration work? And so that leads me into the second half of my presentation, in which I'll be talking about my current research focusing on salt marsh restoration within New England. My goal is to apply the research priorities and opportunities for innovation identified in this interview project to the habitats that I'm currently working in. So I'd like to begin here with some background information on salt marsh habitat. Salt marshes are coastal wetlands that are characterized by grassland vegetation and regular tidal flooding. They have a global distribution within temperate regions, as you can see from the bottom left image, and they have distinct vegetational zones that are determined by the amount of flooding received by each portion of the marsh. Marshes gain elevation through the process of accretion or the accumulation of sediment and organic matter. Accretion and flooding are generally understood to have a positive feedback relationship within marshes, as flooding generally stimulates plant growth and brings additional sediment to the marsh, enabling this vertical growth. To get a closer look at each of these zones of the salt marsh, particularly the species associated with them in New England, it's helpful to look at a profile that shows the amount of flooding that each zone receives. So on the left, we have mudflat, which is almost always flooded and is only exposed at very low tides. In New England, native species of seagrasses such as woodland grass or Ruppia marittima and eelgrass or Zostera marina are found within these mudflat habitats. In the low marsh, which is flooded daily at high tide, the dominant species of grass is Spartina alterniflora or smooth cordgrass. Other species, including slender glasswort, Salicornia depressa, are found intermittently mixed in with this dominant species. Smooth cordgrass can still be observed at lower densities within high marsh habitat, but here the dominant species is Spartina patens or salt hay, which is a generally shorter species of grass that tolerates less frequent flooding, so only during the highest of high tides. Other species observed in high marsh habitat include spike grass or Disticus spicata and salt marsh rush or Juncus gerardi. Salt marsh rush tends to be found within the transitional zone between high marsh and upland habitat, along with species including seaside goldenrod or Solidago sempervirens, and high tide bush Iva frutescens, which is a shrub species and is state listed as a rare species in New Hampshire. I'll speak a little bit more about these species later when I talk about the specifics of my field site, but I wanted to give a little bit of a visual aid about where we could expect to find these species beforehand. So this zonation is important when we consider the effect that sea level rise has on salt marshes. As I mentioned a couple slides back, marsh accretion or vertical elevation gain generally has a positive feedback relationship with flooding. However, increasingly rapid rates of sea level rise are beginning to outpace rates of marsh accretion. In New Hampshire, marshes accrete about 2 millimeters per year, but the rate of sea level rise is approximately 4 millimeters per year, and that results in an approximate annual loss of 2 millimeters of salt marsh elevation relative to sea level. So when this elevation is lost, marsh vegetation is exposed to increased flooding, eventually exceeding the tolerance limits of individual plant species and resulting in substantial changes in marsh hydrology. Ponds within the marsh platform begin to expand into large pools called megapools. And as you can see from this top left image, this one mega pool is at one of my field sites, and it's right next to the road. So the further expansion of this pool could present very significant problems. Marsh habitat also begins to retreat inland. So low marsh dominant species of vegetation begin to dominate former high marsh habitat, and high marsh species begin to move into the former upland transition zone. So when this retreat process meets existing coastal infrastructure, as you can see from actually both of these photos, but especially in the bottom left image, marsh habitat is ultimately lost in a process known as coastal squeeze, wherein it no longer has space to retreat. So these issues highlight a need for restoration techniques that can maintain marsh elevation within an optimal range for native vegetation growth. One technique that directly addresses this need is sediment addition, also known as thin layer placement, TLP, or sediment augmentation. In this technique, relatively thin layers of sediment are added to deteriorating marsh areas to increase elevation. These sediments can be sourced from either dredged or quarried sediments, provided that their grain size matches the composition of salt marsh sediments. Sediments can be added either via spray disposal, as shown in the top right image, and this is generally for larger marsh areas where the sediments are sourced by dredging, or they can be added manually for smaller scale applications, including pilot projects. And the sediments that we got for my particular project are shown in the bottom right image. Sediment addition was introduced in the US Gulf Coast as a beneficial use of dredged sediments, and it has since expanded to other US regions, including the Mid-Atlantic in Maryland and New Jersey, and on the Pacific Coast in locations such as the Seal Beach National Wildlife Refuge. However, implementation of this technique in New England has been very limited to a few small-scale pilot projects. Because of this, coastal regulators in New England need more empirical data that show how marshes respond to sediment addition before this technique can be used on a larger scale. And more significantly, before this can be permitted on a larger scale, because fill within wetlands is really something that we try to avoid. Through the pilot studies that have been done in New England are generally small in scale, the results that they've yielded for vegetation response are very encouraging. So a recent study in the Great Bay Estuary, New Hampshire, was part of a coordinated study between 8 National Estuarine Research Reserves, or NERs, through the US. This is actually a great example of the type of standardized, replicated demonstration project with an experimental design that the practitioners in my interview study promoted. And wonderfully enough, its results in New Hampshire were an integral starting point for my own research here. So in this study, sediment was added to both low and high marsh plots at depths of 14 centimeters and 7 centimeters. As you can see from the photos at the bottom of the screen, sediment addition plots were fully covered with low marsh dominant plants 2 years post-restoration. And this follows recovery timelines that are observed after natural sediment deposition events in New England, specifically in the Great Marsh in Massachusetts. So in these natural sediment deposition events, sediments are deposited either after a storm or a winter ice rafting event. And since recovery is comparable between this manual addition of sediment and natural addition of sediment from the storms, this is generally seen to be something that perhaps we can allow for restoration efforts. Another opportunity for future sediment addition projects is the incorporation of positive interactions between native marsh species. For example, there is a mutualistic relationship between the Atlantic ribbed mussel, Eukensia demissa, and smooth cordgrass, Spartina alterniflora. Many bivalve species are known to positively associate with aquatic plants, and this is why living shorelines projects often incorporate these relationships. So for example, paired oyster and seagrass restoration projects. In salt marshes, filter feeding by ribbed mussels improves water clarity, and their contribution towards nitrogen deposition promotes the growth of cordgrass. Mussels also stabilize marsh sediments through their production of dense fissile threads. So as you can see in this bottom right photo, these sturdy threads allow the mussels to anchor themselves in place. In return, Spartina further filters the water passing through it and provides clumped roots and stems for mussels to anchor themselves in aggregations. Considering these benefits, incorporating ribbed mussels into sediment addition projects could promote vegetation recovery while stabilizing the added soils and helping to build marsh resilience by building on positive species interactions that are naturally occurring within marsh habitats. So with these considerations in mind, I developed 2 primary research questions and associated objectives for my dissertation work. First, how does salt marsh habitat respond to sediment addition, both paired and unpaired with ribbed mussel placement? My objective here is to document the response of vegetation, fauna, and soil microbes to this restoration approach. I also do want to note that soil microbial communities are a relatively new approach to evaluating the success of wetland restoration. Microbes can provide great insight into biogeochemical processes within marshes, but sediment addition studies have not yet documented soil microbial community response, so this is a new area for research. Secondly, how does marsh response to a small-scale pilot sediment addition project compare with the response to a larger-scale sediment deposition? So one that maybe is applied via spray dispersal over a larger area of land and not limited to small wooden boxes. My goal in answering this question is to provide data for coastal regulators in New England to form management efforts that are at a larger or regional scale. So to address my first research question, during the summer of 2023, I established a small-scale sediment and mussel addition pilot project in the Great Bay Estuary in New Hampshire. This project is located within the Adams Point Wildlife Management Area, near the location of the coordinated NERS study, which was conducted a few years prior and had promising vegetation results. There is a very large expanding pool next to the causeway at Adams Point Road, and an area of marsh to the southwest that's a little bit more subject to tidal fluctuations. I established 5 blocks of experimental treatments, both around the pool and in this more tidally influenced location, as shown by the points on the map here. At each block, I replicated the following treatments: 2 control plots, one which was marked only by PVC stakes at each corner, and one contained within a wooden frame, a mussels-only restoration treatment, a sediment-only treatment, and then a paired sediment mussel treatment. Around the pool, these treatments were placed both within the mudflat, so in the pool itself, and in the low marsh surrounding it. And at the more tidal location, only low marsh plots were established because there was no pool of comparable size. Each array of treatments is associated with a reference plot, which is located at a higher elevation of at least 10 centimeters and represents a healthy marsh community and associated plant cover. And those reference plots are indicated in white on the aerial map here. To provide a brief overview of the restoration timeline, in May of 2023, I obtained all necessary permits from the New Hampshire Department of Fish and Game. In early June, I conducted pre-restoration monitoring for all parameters, which I will review in a couple slides. And sediment addition took place in late June. Sediments were sourced from a local quarry and were manually added to sediment treatment plots at a depth of 14 centimeters, consistent with prior research at this site. Before adding sediment, we added a thin feldspar layer shown in the photo to the bottom right to measure accretion and elevation change following sediment addition. The sediment addition was a huge team effort, and the top right photo shows the great attitude of the whole field team despite a very grueling effort, hauling a total of about 2 tons of sediment into these treatment plots. We then planted Spartina alterniflora to the sediment addition plots within the large pool, so the mudflat treatments, because those plots would be less likely to see recruitment of new plants as they're more isolated from nearby vegetation. And in August of 2023, mussel addition took place. We added 10 mussels to each plot, which is consistent with expected natural densities at those elevations, and we tagged and measured each mussel to assess growth and survival. After that, I added mesh to each mussel treatment to exclude predators. And here are just a few extra photos showing the mussel addition work. So we obtained the mussels from the marsh edge closest to the restoration plots in an effort to minimize disturbance when relocating them. And the photo to the right shows a completed sediment and mussel plot within the pool, and it has the predator exclusion netting added. I added mussels that were at a size range between 50 and 80 millimeters because they would still be growing and still likely to reproduce. And so that size range is depicted in the center photo. So both pre-restoration and then for 2 years minimum post-restoration, I will collect data for the following parameters. First, vegetation cover, measured through point intercept surveys to be consistent with prior studies at this site. Hydrology, measured using water level recorders. Pore water chemistry, including pH, salinity, redox potential, and sulfides. Benthic macroinvertebrate community composition, collected through sediment cores and sorted to the lowest possible taxonomic level. Soil microbial community composition, which I'm shown in this photo collecting my little sediment samples for DNA extraction and PCR, and mussel growth and survivorship. So far, my data analysis is very preliminary as it's limited to pre-restoration data. I do, however, have a few graphs showing the species composition of marsh vegetation before restoration, because it's important to understand what species are there prior to sediment addition so that we can accurately gauge recovery. So the graph to the left here shows species composition by treatment at each of the 3 restoration areas. So at the top within the low marsh surrounding the pool, secondly within the pool itself, and then at the bottom at the more tidal low marsh location. Species composition was found to be fairly consistent among treatments. Most pool plots, as you can see from the center graph, are entirely bare ground with a small proportion of widgeon grass, Ruppia marinima, which is shown in yellow. And most low marsh plots are a mix of smooth cord grass, so again, Spartina alterniflora, and then slender glasswort, so succulent species, Salicornia depressa. Reference plots, which are the rightmost treatment in all of these graphs, represent the expected species composition for higher marsh elevations, so what we would be likely to see following recovery should our goals be met. And these generally have slightly higher species diversity. I created a similar graph for pre-restoration vegetation composition by block to see whether the block location or the location around the pool initially differs in species composition. So I observed a similar pattern here with, again, Spartina alterniflora being the dominant species in the low marsh plots and bare ground being the primary cover in the pool. Some exceptions to this pattern do include a higher proportion of widgeon grass within block 3 in the pool, and then a higher proportion of spike grass or Disticus spicata within block 2 in the low marsh surrounding the pool. So this summer, 1 year post-restoration, I will compare percent cover data with these results to determine recovery from sediment addition. I also have some preliminary data on mussel survivorship, as I returned monthly following mussel addition to assess mortality through November 2023, which was around when the first snow of the season came and it was no longer possible to assess. 1 month following initial mussel placement, any mussels that were found to be dead were replaced with a live mussel. This was just an initial attempt to try and make sure that disturbance was not the reason for mussel loss. But after this initial replacement effort, no further replacement took place. A majority of mussels were found to be alive at the most recent site visit. However, certain plots exhibited higher mortality than others. The greatest losses were seen in mussel-only treatments within the pool. So if you look at this pool series right here, the mussel treatments, which are depicted in sort of a coral color, you can see that those treatments experienced a lot of mortality. Other plots that had somewhat high mortality were in the low marsh surrounding the pool, particularly the sediment and mussel treatments. So it's important to note that the lowest and highest elevations were observed to have the highest mussel mortality. And this suggests that mussels may be most successful at mid-range elevations, which would help us refine guidelines for future restoration efforts. The last graph that I want to show depicts the mean plot elevations for each treatment immediately following sediment addition. Sediment addition plots, which are shown in black, are substantially higher in elevation than control plots as intended, and are close in elevation to their associated reference plots, which are shown in white. There is one slight exception. The reference plot is a little bit higher in elevation within the tidal low marsh than the sediment addition treatments. However, this is because there's a relatively steep slope there, so without the plots being too close to one another, it was not possible for the reference plots to be within as close a range as the other treatments. We will continue to monitor plot elevations annually to account for any sediment loss or subsidence that may affect the elevation over time. And the bottom 2 photos here show a sediment addition plot, both within the pool on the left and then in the low marsh to the right. So this sort of just gives a sense of the difference in elevation between the area within the plot and the surrounding marsh. In addition to continued data collection, I have 2 major goals for the next step of this research. First, I will prioritize sharing data with the Great Bay near, the regulatory community, and the public, so that my results get to project end users as intended and can be used to inform future restoration and management efforts. In doing so, I will add my results to existing near data sharing efforts and portals. I also plan to communicate my project goals and progress updates to local community members through events such as community science days, workshops, and other stakeholder outreach efforts. Secondly, returning to my research questions and objectives, I also plan to apply the methods and monitoring metrics used in this project to a larger sediment addition project proposed in Maine in 2025. This is an important next step in scaling up sediment addition research, and it's a collaborative effort with nonprofit organizations such as Ducks Unlimited, federal agencies such as the U.S. Fish and Wildlife Service and the Army Corps of Engineers, as well as university researchers and town representatives. So this type of cross-disciplinary collaboration is, again, something that the interviewees in the study that I did in 2020 recommended for future efforts. So it's really promising to see people in Maine coming together to accomplish this. This sediment addition project is proposed to take place in a much larger 2-acre area of marsh habitat in Wells, Maine. So a dredge of the Wells Beach Inlet, which is shown at the yellow place mark in the second map here, is scheduled to take place through the Army Corps of Engineers this current winter. After the sediments have been dredged, they will be stored in wells until the sediment deposition, which is planned to take place in 2025 at Drake's Island, which is just north of the dredge site. And a photo of the area where the proposed sediment deposition will take place is in the right photo here. This will be a very valuable opportunity to compare smaller scale pilot project results with the first large scale thin layer placement project in Maine. I'm really looking forward to collecting pre-restoration data this summer and to continuing this work in the future. So before I wrap up, I do want to take a moment to acknowledge everyone who has been a part of both my current and prior research. It's the UNH Coastal Habitat Restoration Lab, led by Dr. Greg Moore, and my committee members, as well as other grad students within the lab and all of our undergraduate researchers who are instrumental in getting samples processed and in doing fieldwork. I'd also like to thank partners at the Great Bay National Estuarine Research Reserve, Ducks Unlimited, and the Rachel Carson National Wildlife Refuge. I also want to make sure to acknowledge my colleagues for the SCAS Bulletin article and all interview participants, and the American Honda Foundation for funding this research. And of course, it's important to acknowledge my current funding sources, including the National Fish and Wildlife Foundation, as well as the University of New Hampshire's Graduate School and the School of Marine Science and Ocean Engineering. This is a quick look at all of my references. And I just want to say thank you so much for listening and learning about my research. Please feel free to ask any questions either now or by email. And to check out the Moore Lab website if you're interested in our research. We do a lot of things from salt marsh to mangrove restoration, microplastics, and a lot more. So thank you.
Thank you, Marjorie, for that talk. It's definitely learned a lot, and it's very interesting to see all the stuff that you've been up to. So as Marjorie mentioned, if you guys have any questions, please feel free to unmute, or you guys are more than welcome to put it on the chat. So one question for me, since they're doing the sediment deposition in a larger scale, and when you guys were doing it on a smaller scale, you guys saw that, vegetation came back, like after the 2 years. So let's say if you guys do like this larger scale and you guys aren't seeing the vegetation come in, is there future plans of actually planting some of the native plants to help recruitment of the vegetation?
I very much assume so. I definitely expect to see lower rates of vegetation recovery at that larger scale, just because without as many plants being right around the area of sediment addition, it's going to be a little bit difficult for them to colonize the area. They can grow through. We've definitely seen that. And that was something that was seen at that natural sediment deposition event. I don't know that the partner organizations who are running this project have specific plans for planting, but I don't think it would be outside of the scope of the project. I think they would be willing to do it, especially if the recovery rates just weren't what they expected.
Okay, thank you. I don't know if you mentioned it and I probably skipped it. But I know you mentioned that some of the mussels you had more mortality in on the high marsh, right? Mm-hmm. Is there like a reason why you think that?
I do think so. So I did notice that the low marsh plots that showed some high mortality were pretty much right around the pool and not the ones that were a little bit more influenced by the tides. And I think the issue there is that, and this, once I process the water level recorder data, this will be a little bit more clear, but I know that the fluctuation in water level around the pool is very, very minimal. So even at the highest high tides, a lot of those plots just aren't getting as much water as we expected, which just doesn't leave a lot of opportunity for the mussels to filter feed. So I'm thinking that for future mussel restoration projects, it would probably be necessary for people to really do detailed elevation measurements and really see how much flooding each part of the marsh might get before putting mussels there. And of course, we scoped it out beforehand, trying to see, will they have enough time to filter feed? But sometimes it's just, you go through a tidal cycle and then you really see how much they're getting.
Okay, thank you.
Yeah.
Is there any other questions for Marjorie?
I have one, but I came late, so I'm like not sure if you already mentioned this because based off Anita's question, how you saw higher recruitment in like the smaller plots, have they thought about doing the sediment deposition in like small chunks so that there's still more vegetation surrounding those areas versus like doing a huge area of sediment deposition?
That's a great question. I don't think that that's been proposed. But one thing that we might be doing, and I'm not sure if this is going to happen or not, but based on the timeline of the larger scale sediment deposition, I might be doing smaller scale sediment addition at that site before they do the large scale effort. So in that case, I might be able to see. But, we do see good vegetation recovery. It just, looking at the smaller plots, sometimes, you wonder how much is due to the fact that there are plants right around the plot. But yeah, it's a really good question. I'll be sure to think about it when I'm designing the next stages of the dissertation work.
Yeah, the research that I've read, at least for like sediment augmentation stuff, it takes like 5 to 7 years.
Mm-hmm.
So it's a
Yeah, I'm
long
hoping
time,
we'll
but I
have.
know like
Go ahead.
for nature, that's not that long.
Yeah, no, it's so true because monitoring timelines are generally so much shorter than they need to be. So I'm really hoping, especially for a project of this scale, we'll be able to monitor it for that long to really see, how much is recovering within 5 years at minimum.
Yeah, that'd be so cool.
Yeah.
Just help them out. Thank you.
Any other questions, comments, or concerns? Okay, if there isn't any more questions for Marjorie, I really like to thank you guys for joining today for this amazing talk in order for us to, bring awareness to wetlands and celebrate World Wetlands Day, even though it's like a day late. But, we still get to celebrate it one way or another. And I really hope you guys could be able to, got to learn a little bit more about what Marjorie is doing and the stuff that is happening for restoration on wetlands. And with that, I would like to thank you guys again for coming, and hopefully you guys would join us for our next talk, which would be on March 14th. So just keep an eye out for that email, okay? Thank you so very much.
Thank you.
Bye. Thank you.
Thanks for your